My presentation is about pain sensation analyses in mice brain in a view of future research or treatment in mental disease and its treatment with CRISPR strategy. My name is Shranaya Kumar, and my mentor was Dr. Takuya Osakata from NYU Langone Health and the Neuroscience Institute. So we know that pain is related to a number of diseases and ailments in the body. We know that if we feel something that hurts us, oh, that means that something bad is happening. Or if it continues for an extended amount of time, we know, okay, something is definitely wrong. We need to go to the doctor and we need to go check this out. So now as a community, we know that pain could indicate to us that we may have a certain disease, which includes neurodegenerative diseases. So my research was about pinpointing these regions in the brain that allow us to experience pain, which could then help us to pinpoint what causes Alzheimer's in the brain. And then in the future, we could use genome editing, CRISPR or Cas9 strategy, and these could be used on the regions to protect us from these diseases in the near future. So pain has been an indicator of negative effects on the body in multiple species. So we know that when we experience pain, it's something bad. When mice experience pain, it also can mean something bad. So pain is associated with different chronic diseases, and they can also be linked to social behaviors. So when humans feel pain, we feel depression or anger, or we start to act violently towards other people. So that means that it is associated with emotion in our body. So we know that Alzheimer's is a mental disorder that can cause the degeneration of brain cells. And that means that we forget what we're doing, or we forget different people in our lives. So there have been recent studies that show that the link between dementia and pain can cause patients to experience like increased levels of pain before their medical diagnosis of Alzheimer's. So pain could potentially be used as an indicator of diseases, and they could help diagnose these patients without having the disease progress to that extreme level of not knowing your family members, or forgetting and acting out in certain ways. So this study was conducted in order to connect these pain sensations in mice brain and to potentially create a way to treat Alzheimer's disease with CRISPR strategy. So CRISPR strategy is a technique that is able to edit genes through the precise cutting of DNA. So when you can cut the malicious genes in the DNA, cut that out of the DNA strand, and then the brain or the body will essentially repair that DNA strand to have it more better and not have that malicious gene inside of it. So this overall experiment was conducted on mice brains, and it was divided into 3 separate modules: control versus stress, attack versus defeat, and defeat versus painkiller. These social behaviors, including stress and defeat, can be connected to Alzheimer's patients because Alzheimer's patients often react in violent ways. So that can be like seen in the attack module. So the separate brains, after the social behaviors were done on them, they underwent CFOS staining, which is, as you can see in these images, it's like a green sort of like clusters of green dots. And when you have a significant number of these green dots, they can show expression level in the brain and show these regions are being activated after the social behavior has been conducted in the brain. So some further background is pain in the brain. It's recepted by nociceptors in the skin, and it's sent through the spinal cord to the thalamus in the brain. So the thalamus region is in the middle of the brain. So that's just how the brain communicates feeling and how it shows like this is currently happening to your body. So there's different types of pain. So acute pain is known as short pain. So it's when you step on a Lego and you're like, oh my God, that hurts. So acute pain is when you only feel it for a certain short number of time. However, chronic pain, it's over a long period of time. So chronic pain can be associated with diseases like arthritis. So you feel it for more than 6 months, and it's continuing. So chronic pain has been connected to Alzheimer's. So when Alzheimer's patients feel chronic pain before their diagnosis. They say like they can go to the doctor and this chronic pain can then be associated with arthritis and sorry, Alzheimer's. And then even in Alzheimer's patients who experience chronic pain, it can progress the disease. So currently some drugs that are used to treat pain include NSAIDs, which are non-steroidal anti-inflammatory drugs. So that includes ibuprofen, aspirin, and over-the-counter drugs. And there's also opioids, which include oxycodone and morphine. So we did not use opioids in this study because we know that there's currently like an opioid crisis in humans, and it has a high abuse potential. So instead, in this study, we use ibuprofen to essentially kill the pain that the mice felt. So going back to Alzheimer's, CRISPR and Cas9 has not been used widespread to treat Alzheimer's, but it could potentially be used in order to reduce the plaque that occurs in the brain and then cut that gene out that forms the plaque. And then it could potentially help the patients with Alzheimer's. So in this, first we conducted a control versus stress. So mice were placed into a small tube that causes stress on their bodies. So the regions on the right, we see it in the male mice that were restrained. Those were the regions that were activated that after the mouse felt pain. So the region in the middle, throughout the results, you'll see a region in the middle is activated, and that is the thalamus, which is just responsible for feeling. And it's not significant in this case because it's not really, it doesn't really show like, oh, a different region for pain. It just shows, oh, that mouse is feeling pain. So the region that should be taken into consideration in this study, in this particular module, is the left and right anterior and posterior regions. Anterior and posterior meaning going from the front to the back of the mice brain, the bottom anterior regions, and the top posterior regions. So those are the regions you see highlighted in the diagrams. So then we move on to the defeat versus attack. This particular brain is the brain of a defeated mouse. So the regions that we see highlighted here are the top anterior regions, the left and right anterior and posterior regions, and the bottom anterior region. So these regions were mapped because we see, like in the Olivia images, we see significant CFOS clusters in those regions. And it was after the mouse was placed into a cage with the attack mouse, which we'll see next. So this is the attack mouse who exhibited more violent behavior. So we see that the top anterior regions, the right anterior region, and the bottom posterior region are activated, which is significant because that shows these regions are activated after it is experiencing violent behavior. And in this module, the attack mouse fought the defeated mouse. So that is why the mouse essentially felt defeat and the other one was attack. So in the last module, we use painkiller to see what regions would be deactivated in a way. So the regions that were not deactivated and essentially activated were the top anterior region, the left and right anterior and posterior regions, and the bottom posterior regions. So they exhibited feeling in these regions and caused the CFOS staining clusters to be shown. So overall, we see that the regions of pain that are involved in pain and separate social behaviors like stress and attack were the top anterior regions, the left and right posterior regions, and the bottom posterior regions. So these are the regions that we see were in common with the results in general. So in future studies, these regions could potentially be targeted by the CRISPR strategy. So we know that if there were recent studies that show patients 27 years before they were diagnosed with Alzheimer's, actually diagnosed with Alzheimer's, they said to their doctors they feel pain in certain areas of their body. So we feel that if pain can be located, these specific types of pain can be located using fMRI imaging in the brain. We could potentially use Cas9 and CRISPR in these areas. So these could the specific regions could essentially carry the genes of Alzheimer's, which then means that these genes could be cut out of the brain, and then CRISPR could eventually be used to cure Alzheimer's in a way. However, the scientific world is currently closed off to using CRISPR because of its ethical concerns, because we've seen people have been concerned about using this strategy to basically like edit babies or do like non-ethical things with it. So if these concerns could be solved through ethical committees and other means, this problem could be solved. And then in the future, we could use CRISPR to treat Alzheimer's. So this is the method. The method was done almost entirely by my mentor. So I have little a knowledge about what was done with the mice, but basically CFOS staining was done after the social behaviors happened to the mice. So after the mice fought each other or after it was stressed, it was taken and CFOS staining was done to the mice and to the brains. And then it was seen through a software called Olivia, and that's the images that I use to highlight the specific regions. So these were the references that I used for this paper, and I'd like to thank the program managers and the administration of this program, and like to thank my mentor for his support and guidance throughout this research. It was deeply appreciated, and thank you for listening.